Hello everyone, this is Medical Vishal. Please make sure to subscribe. Let's start with the thyroid gland anatomy. The thyroid gland is situated at the front of the throat, below the larynx. Thyroid gland is a butterfly-shaped, vascular, red-brown endocrine gland situated in the midline of anterior neck. It comprises two lobes that lies on the either side of the windpipe and is connected by an isthmus. An ascending pyramidal lobe is also present sometimes in about 50% of population. Thyroid gland is encapsulated by false fascia and a true fascia. The false fascia is also known as pretracheal fascia, which is a layer of the deep cervical fascia at the front of the neck, whereas the true fascia is also known as the internal capsule, and it is an inner connective tissue covering which cannot be separated from the gland. It forms septae dividing the gland into lobes. Let's zoom in here. So, this is inside the thyroid gland. There are these follicular cells arranged in rings, and here, this cell next to the follicular cells is the parafollicular cell, also known as C cells. The thyroid follicle is actually the main component and is the smallest functional unit here. These are spherical vesicular components of the thyroid gland lined with epithelium. Vesicular because it is filled with fluid, that is the central cavity, or say follicular lumen, which is known as colloid. And this colloid is the place where the precursor of the thyroid hormones, that is thyroglobulin, is stored. So till now, what we did? We learned that the epithelium here contains two cell types. The first one is the thyroid epithelial cells, that is thyroid follicle. And the second one is C cells. The C cells is actually the calcitonin secreting cells. And these are cells of origin of medullary thyroid carcinoma. We'll discuss these cells in detail, but before moving to that, let's check out the space over here in between follicles. This is interfollicular space. By the name itself, it's so clear that it is the space between two follicles. And this interfollicular spaces is filled by reticular connective tissue, fenestrated capillaries, lymphatic vessels, adipocytes, and sympathetic nerves. Now, as we are already talking about the blood supply, lymphatic vessels, and innervations, let's finish it off here. For the blood supply, there are three types of arteries: paired superior thyroid artery of external carotid artery, paired inferior thyroid artery from the thyrocervical trunk, which is a branch of subclavian artery, and unpaired thyroid IMA, that is inferior mesenteric artery, which comes off the abdominal aorta from the brachiocephalic trunk. The superior thyroid artery supplies the superior and anterior portion of the gland and superior parathyroid glands. Repeat with me. The superior thyroid artery supplies the superior and anterior portion of the gland and superior parathyroid glands. The inferior thyroid artery supplies the posterior and inferior portion of the gland and inferior parathyroid glands. Again, repeat. The inferior thyroid artery supplies the posterior and inferior portions of the gland and inferior parathyroid glands. And the thyroid IMA supplies anterior surface and isthmus. Repeat. The thyroid IMA supplies anterior surface and isthmus. Done with arteries. And if not, then I would suggest you to pause the video and write it on a paper once, twice, or maybe thrice. But try to finish the remembering parts wherever possible so that at the end you'll have it somewhere in your memory and you just have to brush it off. Moving to venous drainage. And for that we have three veins: superior thyroid veins, middle thyroid veins, and inferior thyroid veins. The superior and middle thyroid veins drain into internal jugular vein, whereas the inferior thyroid veins drain into the right and left brachiocephalic veins. Again, repeat with me. The superior and middle thyroid veins drain into internal jugular vein, whereas the inferior thyroid veins drain into the right and left brachiocephalic veins. For the lymphatics, it is paratracheal nodes and deep cervical nodes. Repeat, for the lymphatics, it has paratracheal nodes and deep cervical nodes. Now, the innervations. For parasympathetic, vagus nerve, and for sympathetic, there is superior, middle, and inferior cervical ganglia of the sympathetic trunk. Repeat, for parasympathetic, vagus nerve. For sympathetic, there is superior, middle, and inferior cervical ganglia of the sympathetic trunk. Okay, let's get back to the thyroid cells. First, the follicular cells. They are basophilic cuboidal epithelial cells arranged in spherical follicles surrounding the colloid. It has TSH receptor on its surface. It synthesizes, secrete, and store thyroglobulin, which is basically a protein made in the thyroid gland that is precursor to thyroid hormones. Whereas the C cells are large pale staining cells which are found along the basement membrane of the thyroid epithelium and has no direct contact with the follicular lumen. It has calcium sensing receptors on its surface which will sense the calcium from the blood and release calcitonin when required. What's the function of calcitonin? So it decreases the calcium level in blood. For C cells, its function is hormone production and storage in granules. The precursor of calcitonin is procalcitonin, which comes from pre-procalcitonin. Procalcitonin is responsible for proteolytic cleavage of N-terminal and C-terminal peptide to form calcitonin. Now let's understand what is thyroid hormone, how and where it is synthesized, and how it is actually regulated. So for the regulation, we already know that the paraventricular nucleus from the hypothalamus releases TRH, that is thyroid releasing hormone. And this TRH goes to the anterior pituitary via the hypophyseal portal system and gives a positive stimulus to the thyrotrope in the anterior pituitary, which further releases TSH, that is thyroid stimulating hormone. And this thyroid stimulating hormone, as it is an endocrine hormone, therefore will travel via the blood and reaches the thyroid gland and stimulates the release of thyroid hormones. The thyroid hormones, that is T3 and T4, are synthesized by thyrocytes in the thyroid follicles. But how? So let's understand the whole process. For the thyroid hormone to be synthesized, there should be a precursor waiting in the lumen over here in the colloid. So let's bring this precursor in the follicular lumen first. And by precursor, I mean thyroglobulin. In order to understand the synthesis of thyroid hormone, we need to understand that from where and how is this thyroglobulin coming from. You will directly know how this thyroglobulin is formed if you know what thyroglobulin is. So thyroglobulin is a protein. That means it is synthesized in rough endoplasmic reticulum and is made up of amino acids. So let's bring amino acids from blood into the cell, that is follicular cell. But for its formation, there should be a stimulus, and that is from the hypothalamus via TRH, which will give stimulus to the anterior pituitary to release TSH, and it via blood will go to the follicular cells, and there are TSH receptors, which on getting signal will activate G cells. This G cells, which is bound to GDP, will now form GTP and will go to the enzyme nearby, that is this hormone here. This is adenyl cyclase. This adenyl cyclase, which will convert ATP to cyclic AMP, and this will further go to phosphokinase A, and this will go to the nucleus to start transcription of DNA. And we know after the transcription of DNA, what is the result? We get mRNA. Okay, so we get mRNA here, and this mRNA will now leave the nucleus and go to the rough endoplasmic reticulum. Why? Because rough endoplasmic reticulum is the place where translation takes place and protein, that is our thyroglobulin, is synthesized. But this thyroglobulin needs to leave the cell and enter the colloid and wait as a precursor. So for that, packaging of this protein takes place in Golgi apparatus. And after that, this will go to the cell membrane and fuse with the cell membrane and leave the cell via exocytosis. The precursor is ready and waiting for whom? So for that, you need to know what thyroid hormones are made up of. Obviously, thyroglobulin. But is thyroglobulin enough? No. Thyroid hormones are basically thyroglobulin attached to iodine. That means now we need iodine. From where we will get iodine? So from food, like iodized salt. Then this iodine, after getting absorbed into the blood, reaches the thyroid gland. But it needs help to get into the cell. Who will help? Sodium. Sodium will come for help. Sodium is a regular visitor to cells. So it will help iodine to enter the cell. So when sodium is entering, iodine will also enter the cell via this transporter over here. And the name of this transporter here is sodium iodide transporter. But iodine in the cell, is that we want? No. As thyroglobulin is waiting for it in the colloid, we need to send this iodine over there in the colloid. But again, there's a problem. Iodine can't go on its own. It needs help. And for that, we have pendrin, which will help iodine to enter the colloid. As iodine enters the colloid, will thyroglobulin get attached to it on its own? No. They need someone, and that is TPO here, thyroid peroxidase. As it is a peroxidase, it needs H2O2 that comes from a reaction here. Finally, thyroid peroxidase helps thyroglobulin to attach iodine on its tyrosine residue. But again, a question should come to your mind. How we will know whether this thyroid hormone formed here is T3 or T4? Well, it all depends on the attachment of thyroglobulin and this iodine. If while iodination, TPO, that is this thyroid peroxidase, attaches one iodine to thyroglobulin residue, it forms MIT, that is monoiodothyroxine. And if it attaches two iodine to thyroglobulin, it forms DIT, that is diiodothyroxine. And at the end, it will decide whether it's going to form T3 or T4. But all this decision is made in the follicular cell. Therefore, now it's time for this ionized thyroglobulin to leave the colloid, and it is done via endocytosis. As it is leaving the lumen but entering the cell, so therefore endocytosis. There in the follicular cells, lysosomes are waiting eagerly to get attached to this vesicle and cut the DIT and MIT. Therefore, it's time for the decision making, for the big decision. If it cuts two DIT together, like DIT plus DIT, it means the final product will be T4, which is almost 80%. And the remaining 20% will be MIT plus DIT, giving the final product as T3. It is because the half-life of T3 is just about 1 day, whereas the half-life of T4 is about 190 hours, that is almost 1 week. And wherever required, this T4 will get converted to T3 as the affinity of T3 is greater than T4. So this T4 will get converted to T3 wherever required peripherally. And this is done by the peripheral 5' deiodinase or type 2 iodothyronine deiodinase. And finally, T3 and T4 are released into blood via MCT8 transporter, because T3 and T4 are the only lipophilic hormones with charged amino acids and require specific transporters to diffuse across the lipid bilayer. And after coming into the blood, it binds to thyroxine binding globulin, that is TBG, because it can't move on its own in the blood. So it needs a vehicle, and that vehicle is TBG, which is a transporter for T3 and T4 that takes the thyroid hormones away from the cell. The bound fraction of T3 and T4 is biologically inactive. Only the free form is active. If there is increased TBG synthesis, it's called hyperestrogenemia, which is mostly seen during pregnancy and during oral contraceptive use. In this, due to increased TBG, the levels of free T3, T4 will decrease because most of the T3, T4 is bound to this TBG. Therefore, free T3, T4 will decrease in serum, because of which the thyroid gland will think, oh, there is no enough T3, T4, let's make more. Therefore, increasing thyroid hormone synthesis. But if there is decreased TBG synthesis, it's called hypoproteinemia, which is seen during chronic nephrotic syndrome, chronic liver disease, and in steroid use. In this, due to decreased TBG, the levels of free T3, T4 increase in serum. And the thyroid gland will think, there is a lot of T3, T4 right now, so we don't need to make extra T3, T4 or to make more T3, T4. Therefore, the thyroid hormone synthesis will decrease. TBG transports the majority, about 2/3 of the T3, T4, whereas there is another transporter protein synthesized in liver, that is transthyretin, that delivers thyroxine and retinol. It is a negative acute phase protein which decreases during acute inflammation, and this decrease in transthyretin leads to the preservation of amino acids for acute phase reactants like C-reactive protein, procalcitonin, ferritin, hepcidin, and hepatoglobin. And finally, the degradation of thyroid hormones takes place in liver and are excreted via bile. Let's also talk about the inhibition of this whole process. So when there is enough free T3, T4, then they will give a negative signal, or in other words, negative feedback to the anterior pituitary and the hypothalamus, letting them know that we have enough thyroid hormones right now, so you can stop for now. And the last part of the video, that is thyroid hormone function. The thyroid hormone, as we know, stimulates metabolism and growth. Therefore, it increases metabolic rate. Also, it increases oxygen and energy consumption. Plus, it increases thermoregulation. Thyroid hormone affects all over the body, including heart, lungs, nervous system, musculoskeletal system, and even reproductive system. Now remember that only free hormone is active, and T3 binds nuclear receptor with greater affinity than T4. T3 functions 7 B's. You can remember by 7 B's. The first one, brain maturation. It is responsible for brain maturation plus myelin formation plus axonal growth. Next B is for bone growth, as it promotes the synthesis and secretion of growth hormone. The third B is for beta adrenergic effects, that is increased expression of cardiac beta 1 receptors in heart, which increases the contractility, therefore increases heart rate, cardiac output and stroke volume. Therefore, beta blockers alleviate adrenergic symptoms in thyrotoxicosis. The fourth B is for increased basal metabolic rate due to increase of sodium potassium ATPase, leading to increased oxygen consumption, therefore increase in body temperature. Next B, the fifth B is for blood sugar. So there is increase in blood sugar as it stimulates carbohydrate metabolism, therefore increasing glucose reabsorption, increasing gluconeogenesis. There is also increase in glycogenolysis, and there is also increase in glucose oxidation. Next B is for breakdown of lipids, that is increases lipolysis. It also increases lipolysis. The last B is for babies. It stimulates surfactant synthesis in babies. That wraps up our look at the anatomy of the thyroid gland and how thyroid hormones are synthesized, regulated, and function. I hope the concepts were clear and easy to follow. If you found this helpful and enjoyed the video, don't forget to hit the like button and subscribe for more content. And if you have any feedback or suggestion, feel free to drop them in comments. I would love to hear from you. Thank you.